Hey guys, so here we are for Chapter 7. Chapter 7 is all about the cell membranes and structures, basically the borderline between inside the cell and outside the cell. This chapter is, relatively speaking, a little bit shorter than some of the other chapters, so hopefully you can plow through this pretty quickly. I would highly suggest going and watching the crash course video. I think it's called In the Club. Very good. Hank Green does a great job. He's also fairly funny in this one. And then I would also watch the Bioflix little video or movie. That's also very helpful to kind of visualize what we're talking about. So let's go ahead and get started. We'll look at an overview first, and then we'll hit the ground running. Our overview basically talks about how calling it life at the edge. So the plasma membrane is kind of like the dividing line between the inside portion of the cell and the outside. It's basically what separates a living cell from its environment, from its surroundings. It is only about 8 nanometers thick, which means it would take about 8,000 cell membranes stacked one on top of the other to equal the thickness of your textbook page or a piece of notebook paper. So we're talking something that's obviously microscopically small and thin. The plasma membrane exhibits a phrase that you're going to see over and over again. It exhibits this trait called selective permeability, which means not everything gets in and out. It has the ability to restrict what comes across, what enters the cell, what leaves the cell. And in the Hank Green video, he kind of compares it to the line at a fancy nightclub where there's a bouncer who kind of decides who comes in and who gets to leave and who kind of controls the entry and exit. That's exactly what it is. So the plasma membrane is kind of like the velvet rope and the bouncer who decides how and who gets in and how quickly they are able to get into the club. We're going to find out that the plasma membrane is kind of a messy membrane. In other words, it's not just a nice phospholipid bilayer and that's it. It has all sorts of things kind of sticking in and out of it, and we'll see that it probably doesn't look exactly like perhaps what you may have thought. Here's a great picture or model of a protein that's embedded in the membrane, and right through the middle you can kind of see this channel that passes right through the middle. And there's a potassium ion. That little dot is supposed to represent a potassium ion that passes through that channel. So this protein actually is in the membrane, and it allows a passageway for these ions to pass through. And we're going to see that that's actually pretty common. From our previous chapters, we know a little bit of this information already. Concept 7.1 says the cellular membrane is a fluid mosaic of lipids and proteins. And we already knew that phospholipids were the major part of the plasma membrane. And phospholipids, we also already knew, are partially hydrophobic and partially hydrophilic. And there's a word for that. They're called amphipathic. So amphipathic molecules, which are phospholipids, they have a hydrophobic, water-hating part and a hydrophilic, water-loving region as well. And they call this model that we're going to look at of the cellular membrane a fluid mosaic model. And that means we're going to find that the membrane is, for the most part, a fluid-like material. It actually has the consistency at human body temperature of about vegetable oil. So it's not really a solid membrane. It's actually more of a liquid, a thick liquid type membrane. And it's also called a mosaic because it has a wide variety of other molecules and proteins and materials that are kind of embedded in it and on it. It's not like a nice, smooth, clean surface. There's a great explanation if you want to click on this link down here. It's to dummies.com, and it talks about the fluid mosaic model of the cell plasma membrane. So if you want further information, it's right down here at this link. You can click there. Long before we had the ability to see the plasma membrane, long before we had the microscopes that had that ability to go down that small, scientists were doing various experiments with cells and cell parts, and they were chemically analyzing and trying to figure out what this membrane was made up of. And they determined that it was made up of proteins and lipids, primarily lipids, but they weren't sure exactly how it was composed or how it was put together. And eventually they kind of hit upon this idea of this phospholipid bilayer that we've talked about already, because there is liquid inside the cell and liquid outside of the cell. And this phospholipid bilayer makes a nice, stable structure between two aqueous, water-based environments. So here we have that model that we've already seen before. Here we have the phosphoric head, which is hydrophilic, and that is attracted to the water. And then there's the two fatty acid chains that come off that make up the tails that are the hydrophobic parts. And as we can see here, some of these chains are slightly bent. We talked about whether or not they were carbon saturated or unsaturated, hydrogen saturated or unsaturated. And if they were not saturated, oftentimes they had a little bend in one of the tails due to a carbon double bond. And if they were saturated, they tended to be flat, straight. Way back in the early 1900s, 1935, in that area, the scientists who were studying this cell membrane, we didn't have the microscopic technology to actually take a closer look, so they were kind of shooting in the dark a little bit. And what they first proposed, or what two scientists first proposed in 1935, was this sandwich model, a phospholipid bilayer with actually additional layers of proteins on top. And the reason that they thought that there were additional proteins layered on top was when they did experiments with just a pure man-made phospholipid bilayer, it didn't really match what they saw in their experiments with cell membranes. The pure phospholipid bilayer was not as attracted to the water as actual cell membrane material was. So they thought it couldn't just be pure phospholipid bilayer, it must be something else. And one of the other possibilities was proteins that were even more hydrophilic, that were attracted more to the water. So the first idea was this sandwich model with proteins kind of covering or layering the top. Then they determined that that wasn't exactly correct, and they thought perhaps some of these proteins were embedded within the layers, because these proteins also have certain parts that are hydrophilic, that like to be next to the water, and hydrophobic, that like to be away from the water. So in 1972, a further developed idea came about, and this was helped by the advent of new microscope technology also, that perhaps the idea was that the membrane is what they call a mosaic. In other words, it does have a phospholipid bilayer, but there are proteins that are embedded on it and in it. And sometimes these proteins go all the way through. And so the proteins themselves have portions that are attracted to the water and portions that are phobic to the water. And so the parts of the proteins that are phobic would be in the middle of the bilayer, and the parts of the proteins that are philic toward the water would be on the outside. So in 1972, this further model, this enhanced model that we now call the plasma membrane, the fluid mosaic model, came about. So here's a model of what we kind of see now, or what most scientists consider the plasma membrane to be now, a fluid mosaic model where you have, for the most part, you have these fatty acid chain tails on the inside and the phospholipid bilayer heads on the outside. So you have your two layers of phospholipids. And then impregnated within that are these various proteins. And the proteins themselves can have areas that are hydrophobic and areas that are hydrophilic. And notice how some of the proteins pass all the way through, and then we'll find out some of the other proteins are stuck just on the outer edges, and there's a vast variety of materials that are attached to and connected with this phospholipid bilayer. It's not just a nice, smooth, clean layer of material. One of the more modern methods of study that we have now to kind of determine what this membrane is made out of is what we call freeze fracture. Freeze fracture is where they actually take the cell membranes, or entire cells for that matter, and they freeze them with liquid nitrogen or some other material that's extremely cold. And then they basically shatter the cells, or they shatter the cell membranes that they're looking at. And the cell membrane is weakest in the middle of that bilayer. It tends to split apart in the middle of that bilayer, and then they can look at the different edges or the different sides of that bilayer. So freeze fracturing is a technique that has allowed us to break apart the lipid bilayer and look at this cell membrane and gather more details. There's a great photo illustration of this on page 126 of your text, and I'll show you a picture or a diagram in just a moment. And then if you wanted more details, you can come down here. Here's a little link that I added right down here. You can click on that for the information about the freeze fracture technique. Kind of a cool technique, again, using like liquid nitrogen to flash freeze cells and then shattering them apart to further look inside. Here's that photograph and model I was talking about. So keep in mind that this model, when they show a knife and they show this chunk of frozen material, that this is just an illustration. It's a model. It's not going to be exactly like this. But they take a cell, they flash freeze it, usually with liquid nitrogen, they shatter it, and oftentimes that lipid bilayer shatters and splits right through the middle. And then they can actually take, usually electron microscope photographs of what they see inside those layers, and that gives them greater detail and greater information. So here's a more complete model of the phospholipid bilayer and the cell membrane. And again, it's pretty messy looking. It's not a nice clean sheet. And this is a pretty updated model of what the animal cell plasma membrane looks like. And obviously you kind of have a cutaway view. So down here below, this would be the inside of the cell. You can see the microfilament cytoskeleton. And then here's that lipid bilayer with all sorts of proteins stuck into it. And there's some proteins on the outer edges, and there's all sorts of what we call glycoproteins and carbohydrates. And then that connects with all of this extracellular matrix that we were discussing previously. So it's a bit of a mess, but this is a more accurate representation. And the cool thing about models that we use in science is that even though this is probably the more accurate model, other models that we might have had previously are still helpful. So as we gather more information, we change our models and we change our various thoughts and ideas. But older models and older thoughts and ideas can still be useful in certain instances. So in science, we don't always just throw out all the old information. We tend to simply modify and add to it and make it more accurate and usually more complex. I mentioned that this plasma membrane, this cell membrane, is more of a fluid than it is a solid. At body temperature, it's more of the consistency of vegetable oil. So you're going to have some motion. You're going to have some phospholipids within that plasma membrane that can shift and move around. Oftentimes, they'll move left and right. They'll move laterally. And every once in a while, they'll even kind of do a flip-flop, what's called a transverse motion, where the phospholipid that was facing inside the cell flips to face outside the cell. That's a little bit more rare. So here we have a picture or a model. So this lateral movement, kind of like phospholipids shifting left and right, that happens pretty often. But this flip-flopping motion where they might flip-flop facing in or out of the cell, that does occur and can occur, but that type of motion is far more rare. Your textbook talks about how the proteins within the plasma membrane, they also shift and move around just a little bit, not quite as much. They don't drift quite as much because they're larger and they tend to be stuck more in place. But your textbook talks about an experiment where a mouse cell and a human cell were fused together, and so their layers, their cell membrane layers, fused to become one. And there are mouse cell proteins that are specific to the mouse that are different, and human cell proteins that are specific to the human cell. But once they fuse together over a period of time, after 1 hour, they looked at the cell proteins and where those cell proteins were distributed on the cell membrane, and they were pretty mixed together. So that tells you that the human cell proteins kind of traveled and moved around on this cell membrane, and the mouse cell proteins did as well. So these proteins will also kind of shift and laterally drift around, but not as much as the phospholipids themselves. So we already mentioned at normal body temperature, the phospholipid bilayer is fairly liquid, but as temperatures cool, membranes that are made up of lipids can switch from a liquid fluid state to a solid state, just like, say, bacon grease. Bacon grease is made up of lipids, of course. So when it's at a high enough temperature, it's going to be in a liquid state, and you can pour it out of the pan. But then, of course, as it cools down to room temperature, it becomes a more solid material like lard. So these phospholipid bilayers, they can change from a more liquid state to a more solid state, just like a bacon grease can, and that depends upon the temperature. So a membrane that is heavy or rich in unsaturated fatty acids, remember the unsaturated fatty acids have one kinked fatty acid chain in their tail because of the carbon double bonds. They're not completely saturated with hydrogen. So if the membrane is rich in unsaturated fatty acids, it tends to stay in a fluid state longer or more often or at a lower temperature. If the membrane is filled with saturated fatty acids, it's going to be more solid. Again, remember, saturated fatty acids would be more like lard and butter, and the unsaturated would be more like vegetable oil. So the membrane, for it to work, it has to be a fluid state. If the membrane becomes solid, it's not going to allow materials to pass through, in, and out of the cell. So it has to be in a liquid state, and usually that's about the liquid state that you would consider the consistency of salad oil. Cells have a couple of materials that they use to help determine whether or not the cell membrane is in a liquid state and to help keep it sort of in a liquid state so that the cell can do its job. One of those is the steroid that we've discussed already called cholesterol. cholesterol has an effect on the membrane fluidity at different temperatures. When the temperature is warm, such as, say, human body temperature, which is about 37 degrees Celsius, that's right at about 98.6 degrees Fahrenheit, the cholesterol actually helps keep the membrane a little bit more solid. It restrains the movement of phospholipids because at that temperature, the phospholipids are very liquefied, and they tend to move around a lot. And while you need the membrane to be in a liquid state, you don't want it to be too free. So the cholesterol actually constrains these phospholipids at a higher temperature, like say 98 degrees, 98.6 degrees. At a cooler temperature, it actually helps maintain fluidity. In other words, it actually helps keep those phospholipids from packing too closely together, which kind of keeps them in a liquid state and doesn't allow them to solidify. So the cholesterol steroid is actually a very cool buffer. It's like a fluidity buffer. So at high temperatures, it helps keep it a little bit solidified, and at low temperatures, it helps keep the membrane liquefied. So that way that the membrane can work over a larger temperature range. So here you see a model of a phospholipid bilayer membrane. Here it has a lot of unsaturated fatty acids. So remember, the unsaturated ones are not completely filled with hydrogen. They have this kink in the tail because of the carbon double bond. So this means they can't pack closely together, and they tend to be in a liquid state, fluid state, over a wider temperature range, or down to a lower temperature range. Over here we have a membrane that is primarily made up of saturated fatty acid chains. And the phospholipid bilayer can pack together very tightly, which means it's going to solidify at a higher temperature. It's going to be solid more likely than liquid. And then down here we have cholesterol. This cholesterol kind of gets in between the phospholipids, and then it can actually act, like I said, as a buffer. When the temperature is warm, it helps keep the phospholipids from moving around too much. And when the temperature is cool, it helps break them up and not allow them to pack so tightly together, again, keeping the bilayer in a liquid state. The composition or makeup of the lipids in a membrane, cell membrane, is actually due to evolution. Different creatures, different species of creatures have different compositions of their cell membrane, and it's usually dependent upon the environment in which they live. So, for example, animals that tend to live in very cold environments, say, for example, fish that live in the deep Arctic Ocean, very cold environments, well, they're going to have a different composition of cell membrane than animals that live, say, in the desert for the most part. The cell membrane of the fish is more likely to have more unsaturated fatty acids so that it will stay liquid down to a lower temperature. And then that, as opposed to, say, a creature that lives in a hotter environment or a warmer environment, is more likely to have a larger amount of saturated fatty acids because it doesn't have that same concern. There are even living creatures that have evolved the ability to change the composition of their lipid bilayer based on their environment. So there, for example, are a lot of plants that have a warm growing season, but then in the winter months they live in a very cold climate. And a good example is like winter wheat. And this plant actually changes the percentage of unsaturated phospholipids in its cell membrane in the fall. So because the temperature is going to be getting colder and colder, this plant responds by upping the amount or percentage of unsaturated phospholipids in its cell membrane. That way the cell membrane can stay in a liquid state even as the temperatures start to fall. Other than these fatty acids, the phospholipid bilayer, the other major portion of the membrane is the proteins that are embedded in it. And there's a wide variety of proteins that we're going to talk about, and this kind of makes it this mosaic that we talked about, because a mosaic is, say, made up of a lot of different pieces, maybe a different broken colored tile and things like that. Well, this plasma membrane mosaic is made up of a lot of different types of proteins embedded in it and on it. And while the phospholipids, they make up the main portion of the membrane, it's actually the proteins that are embedded in it that determine its function and do most of the actual work and the actual jobs of the membrane. Of these proteins that are part of the cellular membrane, some of them are just called peripheral proteins, and based on their name you can probably guess Those are kind of outside edge type proteins. They're bound to the surface of the membrane, but they don't actually go all the way through the membrane. So peripheral proteins are stuck to the surface in some way. Oftentimes they help anchor the cell to the extracellular matrix and things of that nature. And then the other ones are called integral or integral, depending on how you want to pronounce it, integral or integral proteins. And these actually penetrate the hydrophobic core of the bilayer. In other words, they pass all the way through, and so there's usually a portion sticking outside of the cell and inside of the cell. And these integral proteins are also called transmembrane, trans meaning across, so they cross the entire membrane. And they usually have a hydrophobic region to themselves, that it's going to be toward the inside, and a hydrophilic region, which is toward the outside. The hydrophobic regions of these proteins usually consist of a couple of stretches of nonpolar amino acids, so that nonpolar usually makes it hydrophobic, and they're coiled in what's called alpha helices. Remember, there was alpha and beta, and the alpha was kind of the twisted helix, and the beta was kind of the pleated sheet. Well, these tend to be the alpha helix, and so this nonpolar region is going to be toward the center of the membrane, away from the liquid environments inside and outside the cell. Here's a quick model of a transmembrane protein, or also called an integral or an integral protein. It goes all the way through, and it's going to have some outside edges that are hydrophilic, and then here's that helix, alpha helix portion inside that is nonpolar and usually hydrophobic. These membrane proteins have 6 major jobs or 6 major functions, and we're going to not go into a whole lot of detail about all 6 of these, but we do need to kind of know all 6 and maybe an example. And the book does a great job of giving you a diagram or a picture for all 6 of these, and I'll show that to you in just a moment. So here are the 6 major functions or jobs of these proteins that are stuck into this cell membrane. One of them is transport. We're actually going to talk quite a bit about transport. Then the second one is enzymatic activity. Remember, an enzyme is something that speeds up a chemical reaction or speeds up a chemical process. One of them is called, number 3 is called signal transduction, and that's basically a messenger. We'll talk about that. Number 4 is cell-to-cell recognition. And number 5 is intercellular joining. So that is kind of getting to how the cells stick together and more of the skeletal or the cytoskeletal portion. And number 6 is attachment to the cytoskeleton and the extracellular matrix. And again, that's going more to the physical portion of attaching cells together or attaching cells to all that extracellular matrix that's outside of the cell that we talked about in the previous chapter. On page 129, if you want to take a look in your textbook on page 129, here's this figure. It's, I think, 7-10 or 7.10, and it shows all 6 of the functions. So right up here at the top, transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and then attachment to the cytoskeleton and the ECM. And we'll look at each one of these a little bit more detail, but on page 129 you can get a nice summary of all of this. So here are the first 3 functions. One of the functions of the proteins can be transport. And so here's 2 different models of transport. We're actually going to talk a little bit later about these models of transport. So that is basically moving materials in or out of the cell. The second one is enzymatic activities. Some of these proteins act as enzymes, and they help speed up chemical processes inside the cell. And so this is going to be representative of enzymatic activity. The next one is signal transduction. Signal transduction basically means that floating outside of the cell, there could be particular molecules that we're going to call signaling molecules. And when they attach to this protein, that causes a change in the protein that then allows it to signal in some way or send a message in some way to some other part of the cell or to some other protein to allow another process to occur. So this is basically like a mailman or a messenger. He's getting a message from one end, passing that to the other end so that that information can be passed along and some new activity can take place. Here are the other 3 functions. One of them is called cell-to-cell recognition, and this is generally a short-term process that we'll discuss just a little bit, where cells basically recognize each other based on the different proteins that are embedded into their cell membranes. The next one here is intercellular joining. This would be more of a permanent process that kind of physically attaches one cell to the next. And then the last one is involving the cytoskeleton and the ECM. And again, this would be more of a physical process where these proteins are attached to the cytoskeleton within the cell, and they're also attached to the extracellular matrix outside of the cell. So it provides some structure and support to all of this material, because there is no bone or skeleton to the cell, but all of these little parts we discussed make up the structure and function that hold it all together. Let's go back for a moment to this cell-to-cell recognition, because this turns out to be very important, especially when you're talking about the immune system and your body fighting off diseases, and if you talk about blood types, or you discuss, say, like organ transplants, and why some people have trouble with organ transplants when their body starts to reject the organ that they were given via a surgery. So that all has to do with cell-to-cell recognition. So let's take another quick look at that. Cell-to-cell recognition has a lot to do with molecules that are attached to the outside of the cell membrane. And a lot of times the molecules are carbohydrate molecules. Sometimes they're carbohydrates combined with lipids, and that's called glycolipids. Remember, the gly part kind of tells you it's a carbohydrate. And then sometimes they're carbohydrates attached to proteins, which we've actually already discussed before, and those are called glycoproteins. So usually on the outside of the cell membrane, you have these various carbohydrates, glycolipids or glycoproteins, and they kind of are a signaling mechanism to other cells. So this is basically the way that cells recognize each other. It's almost like, if you can imagine, if you go walking your dog in the neighborhood and you meet someone else who's walking their dogs, inevitably the dogs want to sniff each other, right? So they kind of they greet each other, or they recognize each other by sniffing each other. Well, this is kind of like cells sniffing each other's rear ends. So these cells recognize each other based on these various glycoproteins or glycolipids that are attached to the cell membrane. It's kind of like their way of signaling or recognizing each other. The carbohydrates that are attached to the outside of the plasma membrane varies among the species. So, for example, a mockingbird cells and a human cells have different carbohydrates attached to the outside of the cell. And even among individuals, like, for example, if you look at all humans and you look, say, at blood type, we have different blood types, A and O, and A negative and AB positive. Well, that has to do with the different types of carbohydrates that are on the outside of the blood cell membrane. So if you give certain blood types to certain people, there's a negative reaction because the body doesn't recognize those blood cells, or it sees them as an invader. So there are different cell types even within the same species, and sometimes even within individuals. You can have different carbohydrates that are on different cell membranes. This really comes into play when you talk about disease and the immune system. For example, your book talks about a study in which there are some people who have a natural ability to block HIV. They are naturally immune to the HIV virus, and they have been exposed to the virus, but they never get HIV and they never get sick, and their body basically has the ability to block that virus from entering their cells. And as it turns out, on most people, the cells have a couple of different receptors. So they name these receptors right here, CD4 and CCR5. And for the HIV virus to actually attach and enter the cell, you have to have both of these receptors. So the virus is actually kind of looking for both of these receptors, and that's its entry point to get into the cell. But on some people, they do have one of the receptors, the CD4, but they do not have the other, the co-receptor, CCR5. So this person, see, the virus is trying to attach, but because this person's cells lacks one of the co-receptors, the virus may be within the body, but it never actually is able to enter the cell. So this person does not get infected, is resistant. So the thought is perhaps there's a way to develop a drug that in most normal people can block this co-receptor. So there's no way to get rid of this co-receptor from your cells, but what if we can make a drug that locks onto this receptor and basically blocks the virus from attaching to it? Well, that would give you immunity just like some people have naturally. The last thing we want to talk about in Concept 7.1 is how do these various proteins and carbohydrates get on the membrane? Well, it has to do with how the membranes are created. Remember, we talked about the endoplasmic reticulum, which is a membrane-creating part of the cell, and then we talked about the Golgi apparatus, which takes what the ER makes and then further develops it and then releases it. So it has a lot to do with the endoplasmic reticulum and the Golgi apparatus. And when these membranes are made, they have an inside and an outside. So think of it like a coin. It's the same thing, but it has two different faces. Well, these membranes are made as a whole, but they have an inside and an outside face. And the location of the proteins and the carbohydrates on the inside or outside is determined when the membrane is built by the ER and the Golgi apparatus. So a picture is always better than words. So let's take a look at a model or a picture to kind of understand this idea. So here we have the endoplasmic reticulum. And remember, it is creating membrane, and it's basically squeezing off portions of this membrane. And you can see kind of the blue and then the peach or brown color here. So as the ER creates membrane and then releases these vesicles, there's different types of proteins and glycoproteins and glycolipids that are either inside or attached to the outside of these vesicles. These vesicles usually then fuse to the Golgi apparatus, and the Golgi apparatus adds materials, adds proteins, adds other materials as it travels through the Golgi apparatus. And then the vesicles that leave the Golgi apparatus, notice there's some material inside, some connected to the outside. As that fuses to the outer wall, the plasma membrane, what was the inside of the vesicle becomes the outside of the cell membrane. And what was the outside of the vesicle becomes the inside of the cell membrane. So that determines where these different proteins and carbohydrates and molecules are going to be, whether or not they were originally on the inside or the outside of the vesicle. That determines whether or not they're going to be on the inside or the outside of the cell membrane. Let's take a pause right there. We don't want to go too long. We want to break this up into small sections. I know you guys probably don't feel the same way because I am a complete science geek, but when I study this stuff, I am just amazed. Not only am I amazed that this stuff actually occurs, but I am amazed that we have the ability to learn about it and know about it. That's pretty cool. At any rate, I'm going to take a break right here. I thought I was going to go on, but I think I'll take a break. I'm going to go get some iced tea. I'll be back. You come back too.